Assalamualaikum warahmatullahi wabarakatuh. Hello everyone, we are from group 8. Want to explain you about the transgenic animal as a form of assignment from a bioscience course with a supporting lecture, Professor Dr. Muktiningsih Nurjayadi MSI. And for our group members, there are Mahardhika Kurniaputra, Nuraeni Solihat, and Nurhalizah. And for the topic outline of our discussion today are introduction, evolution of the transgenic animals, the cons, and the conclusion.
Okay, the first is introduction. What is transgenic animals? A transgenic animals refers to an animal that is altered by the starter of recombinant DNA through human interferences. The term transgenic animal refers to an animal in which there has been a deliberate modification of the genome. The material responsible for inherited characteristics, in contrast to spontaneous mutation, foreign DNA is introduced into the animal using recombinant DNA technology, and then must be transmitted through the germline so that every cell, including germ cell of the animal, contain the same modified genetic materials. That transgen is mean a technique for insertion of cDNA, which is created by specific mRNA or messenger ribonucleic acid into a cell. The technique applied to create transgenic animal by the direct manipulation of DNA have negative effects of human welfare, society, and environment. Okay, the second is about evolution of the transgenic animals. The history of transgenic animals begins from two biologists, there are Watson and Crick, discovered the DNA in the 1950s, and from that time the basic concept of the transgenic animals by manipulating DNA have come. During the 1970s, the first chimeric mice were produced by Bernstein that the cells of two different embryos of different strains were combined together at an early stage of development, that is, 8 cells, to form a single embryo that subsequently developed into a chimeric adult, exhibiting characteristics of each strain. And then, in early 1980, Gordon and Rado first experimented with the gene addition with the aid of technology, namely microinjection, and from that time a new waywardness was come in genetics and molecular biology research field to drive forward. The DNA microinjection, the first technique to prove successful in mammals, was first applied to mice and then to various other species such as rats, rabbits, sheep, pigs, birds, and fish. And two other main techniques were then developed, thus of retrovirus-mediated transgenesis technique by Janis at 1976 and embryonic stem cell-mediated gene transfer by Gassler et al. At 1986. Okay, the next is mechanism of production the transgenic animal. The first step is identification of the desired gene or DNA and promoter sequences. And then the desired gene is then directly housed into the nucleus of a donor egg by following apt method. Then the engineered cells are then implanted into the host animals. Then the development of the desired cells within the host enable to provide the desired gene that has been stably and heritably incorporated into the newborn offspring's DNA. Unless the gene is then able to continue its function in the new environment. The complete mechanism as shown in the picture on the left.
Transgenic animals in Indonesia. Transgenic animals are animals that have undergone engineering of the arrangement of their genetic material so that animals or plants are produced that have the characteristic desired by humans. Transgenic technology in animals can be done through several techniques. For example, by injecting DNA fragments micro into eggs that have undergone fertilization. The purpose of this technology is to improve products from farm animals such as meat, milk, and eggs to have high beneficial values. Based on the government regulation of biosafety of genetically modified product, Article 1, Paragraph 7, which states genetically modified product or modified organism, hereinafter abbreviated as PRG, are living organism. Their parts and/or processed products that have a new genetic makeup from the application of modern biotechnology. Then, modern biotechnology is the application of genetic engineering techniques that include in vitro nucleic acid technique and cell fusion of two or more types of organisms beyond taxonomic kinship. And GM animals are animals that result from the application of genetic engineering techniques that most or all of their life and are on land. GM animal origin materials are all materials produced from GM animals and can be future processed for human and other purposes. And then, why transgenic animals are still produced? Okay, there are two reasons why GMO animals, especially in Indonesia, are still in production, including one, some transgenic animals are produced to have specific economic, and two, other transgenic animals produced as disease models are genetically manipulate animals to show symptoms of disease so that effective treatments can be studied. And then, example of animal transgenic in Indonesia. 1. Transgenic pig. Pigs are one of the animals that are GMO gene. Transgenic in pigs is expected to improve the quality of pigs and can reduce environmental pollution. This is because pigs usually excrete feces that contain pollution, so that weather products brings many diseases. Pig GMO is done so that pigs can absorb phosphorus so that the wastewater product is like other water. And in addition, in China, already development low-fat pig GMO. This genetic engineering is done in the hope that low-fat pig will have direct bodies so that they have cheaper livestock costs and do not suffer in. The pigs were given a gene capable of regulating internal temperature by burning body fat. And 2. Transgenic fish. GMO in fish is a genetic engineering technique that aims the produce farmed fish fry that have superior properties for the development of fish quality. Transgenic of dairy cows. Transgenic dairy cows can produce gene with casein production of more than 13%. High casein milk production is expected to be used for making better quality cheese.
Okay, next are the cons of the transgenic animals. The cause of the transgenic animals could threaten the growth of the original animals' varieties by spreading their transgenic genetics, resulting in crossing of gene exchange with the original one, and resulting in the animal turning into a completely transgenic animal, or in other words, the transmission of its elements properties to non-transgenic animals. Behind the various benefits provided by its biotechnology, there are also negative impacts that follows in the form of psychological and the physiological and the biological health of the animal. Animals do not reproduce normally, and there is a risk of cancer in humans who consume the biotechnology produced animals. Okay, the next is conclusion. A transgenic animal refers to an animal that is altered by the starter of the recombinant DNA through human interferences. There are many transgenic animals in Indonesia. For example, there are transgenic pigs, transgenic fish, and transgenic dairy cows. Behind the various benefits provided by its biotechnology, there are also negative impacts that follow, in the form of psychological and the physiological and biological health of animals. Animals do not reproduce naturally. There is a risk of cancer in humans who consume biotechnology-produced animals. And there is the reference of our presentation today. And thank you for listening our presentation. Wassalamualaikum warahmatullahi wabarakatuh.